Welcome everyone, and for the second part of the workshop, which is dedicated to Western blot technique. Please be not hesitate to send the questions in the chat, and our meeting today will end with the questions and answers session in the end. And there are no stupid questions. I will appreciate all your questions and will try to answer as best as I can. I'm not certain how many of you who were present on part 1 are here today. So just a short introduction about Agrisera, established over 40 years ago. We are based in Sweden, in this beautiful landscape of northern Sweden. There's our antibody production farm. We have over 80 employees and are part of Olink since 2020, and now for 2 years we are part of Thermo Fisher. Our company is also ISO certified. And Agrisera is the provider of antibodies for plant proteins for over 25 years, and we have been honored to receive the prize last year, the Supplier Succeeding in Plant Science, and this is thanks to the plant community, which is helping us to suggest which antibodies to develop, and also develop antibodies not only to plant proteins, but also hormones, ELISA kits for plant hormones, epitope tag antibodies. We are also offering high titer secondary antibodies, and our products are cited in over 10,000 publications. And I also hope that the content of this workshop will help you to obtain publishable results in the future. So let's dive into what is going to happen today. Part 1 of this workshop covered the Western blot protocol until protein transfer. And for those of you who were not present during part 1, the recording is available on Agrisera website, and today I will cover remaining parts of the Western blot protocol, and also I will include a part about interpretation of results, which, as you know, can be sometimes a bit difficult. Reminder of what we talked about last week. Antibodies are Y-shaped molecules which are having an antigen binding domain in the changeable region, and the antigen binding domain is a part which is of importance when antibody is binding to a target protein. And I want to just remind you that in the nature techniques, as Western blot immunohistochemistry, when proteins are denatured, antibodies are binding to so-called linear epitopes. And there are also other types of epitopes which cannot be predicted in any way, which are brought by protein fold in 3D structure of a protein. And these epitopes are recognized in techniques where antibodies are binding to native proteins, like in immunoprecipitation, CHIP, or Western blot conducted in native conditions. And please remember to save yourself effort in the lab. Check if the sequence an antibody is made to. Usually it can be 5 to 15 amino acids or partial or full-length protein, if that sequence is found in a protein you're aiming to detect. This is a crucial point which will decide your success later in Western blot. Without that knowledge, please never use any antibody, because you can predict based on sequence if antibody is going to work on a protein from the species you're working with, as well as in the application. I have covered it in more details in part 1, but this is so important, so I need to point it out again. And now we are starting second part of Western blot protocol. We are blocking the membrane, and usually the blocking is done either in the cold room or in room temperature. But what decides that? Do you know? And also, please don't use these tweezers, like the tweezers like on a picture don't have flat ends, and they may damage the membrane. but the blocking can be done, of course, 1 hour room temperature or 4 degrees overnight. And it is actually protein antibody pair dependent. So you cannot predict it in advance unless you actually conduct it and receive the data. And please note that both membranes, PVDF and nitrocellulose, can be actually dried after blocking and used 1 to 2 weeks later. It has happened to us that the antibody was not available, the procedure was already conducted, so not to do the whole procedure again, we dried the membrane, protein side up, and used it 1 week later when the antibody arrived. You will receive this presentation, so please don't worry. This material will be available for you, and each slide has this box with the specific information as well. And membranes, as I mentioned, can be stored for later use. And this is something what makes work much easier for us because we are preparing the membranes in advance and store them for 6 to 12 months, dried, protein side up, in plastic bags in room temperature. And both membranes, PVDF and nitrocellulose, can be stored this way and used later. And when you think about it, the proteins are much more secure on the membrane compared to the extract in the freezer, because some proteins may, as you know, degrade during storage in the freezer. So the way to store precious samples, it is on the membrane after transfer is done. And membrane blocking reagents, I have listed them in the order or of how commonly used they are for plant proteins. So 5 to 10% non-fat milk. Then we have very well working commercial blockers. PVP40 is a non-protein blocker. It's a reagent which is used during fractionation studies. And BSA is, as you see here, down the list. Because I have experienced that in many cases BSA was not so efficient for blocking of membranes where plant proteins are separated. And then in the end we have gelatin. And now look at this result. The lab returned this result and told me that the antibody is absolutely non-specific. Look how many bands we are getting. Okay, but please check carefully. What are they using for blocking? Concentration of a blocking reagent is low and the blocking time is too short. So in this specific case, I asked them to extend blocking for 1 hour and then the result was improved. So in this case, it was not an antibody which failed, but the procedure was not optimized. There can be other situations like this one when blocking over 1 hour is not sufficient. So in this case, the blocking had to be extended overnight. So please remember that for some protein antibody pair, the blocking at 1 hour at room temperature will be enough, but for some, the blocking needs to be extended for overnight. And this is, as I said, decided by protein antibody pair. This is what is deciding the blocking length. And you need to check it experimentally. And please, a note for the future, if you are using avidin streptavidin detection, milk cannot be used as a blocker because there will be an interference. And I am aware that in many cases in Western blotting, there can be a lot of unspecific background. And there is actually a way to remove it. If you need, by some reason, there's too many bands, but then you see a target band, but amount of non-specific bands is very high, then you can do so-called adsorption. So if you have knockout mutant, like in this picture, where target protein is absent, you need to prepare a membrane with extract with knockout mutant and incubate the antibody with that membrane, which is lacking your target protein. The background antibodies will bind and will be depleted, but the the antibody for the target protein will remain. So first you incubate your antibody with a membrane prepared with samples from knockout mutant, and afterwards you use that antibody for detection on a membrane of your interest. And this is so-called adsorption procedure, which can remove unspecific background. Researchers who are using primary antibodies are aware of the fact that if there is a background with each subsequent reuse of primary antibodies, the background signal will decrease. So you are already using an absorption method. And in this case here, there is for your future reference, a protocol and a link where you can find exactly step-by-step how to do. Membrane blocking and stripping. I received the question why, after stripping of a membrane, which is very harsh procedure, blocking a background signal can increase. And why? So if you are using a stripping of a membrane, you can also strip the target. And Western blot is about signal to noise ratio. So if you strip the target, there will be less possibility for the primary antibody to bind to the target, and therefore the strength of the background signal will be increased. And this procedure, stripping of a membrane, I understand that you want to reuse it, but in many cases it can lead to deterioration of results, especially if proteins are of low abundance. It is very tempting to compare antibodies with each other. However, I would definitely not recommend it. Antibodies are a little bit like people. It's not possible to compare them between each other. Because if you are using an antibody, which is to proteins of high abundance like actin, the signal will be visualized very promptly. And if you in parallel are detecting a protein of low abundance, you cannot compare intensity of a signal between these two proteins. And the protocols to develop the reaction may also be different. As you remember from previous meeting, the abundance of a protein can be checked in the PAX database. So please do not be tempted to compare antibody performance between different antibodies, because it does not work that way. Of course, there are also questions if monoclonal antibodies are better than the polyclonals, and I can reveal here that, of course, it cannot be compared that way. As you remember, monoclonal antibody bind to one epitope, polyclonal antibodies bind to a wide range of epitopes. And their specificity will be different, and there are different factors which will determine successful antibody binding. Therefore, please do not be tempted to compare antibodies between each other. There is also a concept of loading control. However, I would like to, probably you are using maybe any of those proteins which are very commonly used as loading controls, tubulin, actin, rubisco sometimes, histone. However, please remember, the purpose is to assess equal protein load, but housekeeping protein levels can change depending upon experimental conditions. So you cannot choose a priori that actin is always good as a loading control. Actually, that loading control should be chosen not based on publication record, but on mRNA profiling. This is how you should decide which protein is not changing in the experimental conditions. I would like to show you a result where I will show you a result later where you will see that actin is actually changing depending upon experimental conditions. Most commonly used for primary antibody incubation is TBST. Some laboratories are using PBST. But I would like to share this result with you, where by some reason in this protein antibody pair, as you see here, PBST buffer gave a cleaner result and less background. And please remember, if you work with phosphor-specific antibodies, then the PBS buffer should not be used. And the next step is our primary antibody incubation, and it can be done in different ways depending upon protein antibody pair, and also, of course, the time is the essence. So if you would like to, if you have precious samples, you cannot prepare many membranes, you can actually do parallel primary antibody incubation. And this is one membrane which has been incubated with 3 primary antibodies at the same time. It was the samples were very few, and therefore this approach. This is possible when you know the primary antibody and you know in which dilution it can be used. So it is simply to mix, instead of preparing 3 different membranes, you can mix 3 primary antibodies with each other. However, you keep in mind that target proteins which are detected should be at least 20-25 kilodaltons apart from each other. And that approach will save you the time. The second approach is also a parallel incubation, but look at this, what happens here. Primary, secondary antibody and blocker are used in one incubation at the same time. And this is an approach which can be used for proteins of high and moderate abundance, and also for antibodies which you know how they perform. And of course, this will save you a lot of time, because instead of several hours, this reaction has been developed within 60 minutes. And a third method to speed up your protocol is using antibody which is primary antibody which is conjugated, directly conjugated to an enzyme, horseradish peroxidase, alkaline phosphatase, or a fluorescent dye. However, here you see an example of Rubisco large subunit primary antibody, which is conjugated to the enzyme and is detecting Rubisco subunit in different plant samples. Keep in mind that directly conjugated primary antibodies can be used for proteins of high abundance. If you have a purified primary antibody, you can use this approach. There are kits to label primary antibodies very fast with different labels. However, if this technique can be used, is decided upon with abundance of your target which you're trying to visualize. It will simply work better for proteins, for detection of proteins of high and moderate expression. And to develop Western blot, we need to use secondary antibodies. And secondary antibody is is a protein which looks exactly, is built exactly the same as primary antibody. So we have antigen binding domain and a constant region, and secondary antibodies can come from those different hosts, and depending to in which species your primary antibody is made, you need to choose appropriate secondary antibody. And secondary antibodies, one would think that there's no difference between them, and they are enhancing the signal, which is especially important for proteins of low expression. So here we are using an enhancer, which is a secondary antibody. So if a primary comes from rabbit, secondary needs to be anti-rabbit. And there is actually a difference between secondary antibodies, as I can show you here. And when you're purchasing secondary antibodies, you are probably very much focused on price. I would like to show you that one should actually focus on in which dilution the secondary antibody can be used. because secondary antibodies can be used in 1 to 5,000 or 1 to 25,000 and more. And please do not incubate secondary antibodies longer than 1 hour at room temperature. And here you can count for how many more blots you can use a secondary antibody, which can be used in dilution 1 to 25,000. And of course, then with such dilution, you can use 1 ampule for your whole PhD. And that's good for reproducibility of results. So please keep this in mind for the future. And here a quick example. Which secondary antibody you would like to choose for Western blot? Here is the standard goat anti-rabbit, horseradish peroxidase conjugated antibody, or the antibody which have been adsorbed and has minimal cross-reactivity to bovine, human, goat, and mouse rat IgG. So think for yourself which antibody you would choose for simple Western blot on plant extracts. And here comes the answer. It would be the first one. Adsorbed antibody would also work, but of course it's not necessary because you're not having any of these IgGs in your sample. Therefore, such as adsorbed secondary antibody is not necessary to use. Now we are coming to the exciting part of target visualization. I have been working with ECL and developing my first blots using X-ray film. Nowadays you are using other machines, sophisticated machines and CCD cameras. You're not dealing with reagents, and the visualization of a target protein is the last step of Western blot. And we have a secondary antibody which is conjugated with, in this case, in this example, conjugated with an enzyme, but it can also be a fluorophore. So most commonly used is chemiluminescence, which is used for reaction visualization. We can also use fluorescence, and we can use also chromogenic detection. And results can be different depending upon which method is used. So when we have a chemiluminescence, then we are using chemiluminescent reagents, which are mixed before applying on a membrane. And of course, we need CCD camera for visualization of result. And here it can happen that we may use too sensitive ECL on protein of high or moderate abundance. Usage of extreme low femtogram detection reagent is not really necessary. So in this case, the background comes from too sensitive detection reagent which has been used on this blot. So this is not an antibody fault, it is a too sensitive reagent. So please check before you do your Western blot if your protein is of moderate or high abundance. Here we have two reagents, and for protein of high abundance, we need to choose a reagent of lower sensitivity. And of course, we have advantages connected with chemiluminescent detection. It is a sensitivity and quantification, but the signal will fade much faster. And sometimes there will be background. You are probably familiar with these types of blots. And I would like to provide you with a checklist what to do if such a background will appear. There can be different reasons for it, because it can be also sometimes it can be interference from a plastic which is used to wrap the blot if you are using this method. But also the reason for a background can be that there is not enough of a target protein to detect, and therefore everything else is detected because there's no target which is coming first, because the signal from the target should be visualized first. And in chemiluminescent detection, we have this issue of these unspecific bands. And if your blot is looking like this, then you should actually go back and review your procedure and check that maybe the transfer of the protein was not working correctly, and therefore there is no target band to visualize on your Western blot. And in some cases, we have so-called ghost bands. You are probably familiar with them. They look like ghosts, and the color is white instead of black. It means that's too much of everything, too much of a protein, and too much of primary and secondary antibody. So to simply adjust it, the protocol needs to be optimized. At chemiluminescent detection, I have received a question that something went wrong, and should I redo a whole procedure? And the answer is that one can redo that. You don't need to redo the whole procedure and prepare a new membrane. You can just wash the membrane and develop it again within the same day. Maybe you mixed the reagents wrongly, so this is still possible to save. And fluorescent detection is very good if we want to detect, for example, two different targets, like here, which can be visualized on the same blot with a fluorescent detection. And we can use it for multiplexing, and the signal is staying on the membrane much longer. We will, of course, need a CCD camera. And as with each method, there are advantages and disadvantages with fluorescent detection. When I analyze the publications and troubleshoot, I see that chemiluminescent detection is still most commonly used for visualization of proteins in Western blot. And a third method is colorimetric detection, and that method is cheap. You don't need any CCD camera. However, this method is not suitable for proteins of low abundancy. So please keep this in mind, that you can obtain results which you can take the membrane and include it in your lab book. But this method is not suitable if you work, for example, with transcription factors, because sensitivity of this colorimetric method is way too low. So please keep it in mind when you're planning your Western blot experiments. So detection can be done in 3 different ways, and the outcome will be, of course, different and hopefully publishable. And it is very important when we are working with protein visualization to assess molecular weight of the target protein in a correct way. And there is a method which we are using, which is we are using markers. We are mixing the markers, the colored markers. We are mixing with markers with IgG binding site. And this allows us for visualization of a target protein directly during recording of the signal. The magic marks have very good span of molecular weights. And of course, there are other methods, but it's very important that target protein molecular weight is assessed correctly, and that you are doing it in a standardized way, because this is going to be very important for interpretation of results later. In some cases, antibodies to tag proteins are giving us a hard time, and there's no signal with one and the same epitope tag antibody, which is working for one epitope, one tagged protein, but not for the other. And I have prepared a checklist for you what to do if there is such a situation. because I received such question. Most important is expression level of your tagged protein. This can actually be a reason why there is no detection and no visualization of epitope tagged protein. And I see here that I need to switch to another presentation because the slides which I wanted to show you are actually not included. So let me change. I apologize for this. No, that's still the wrong one. I'm very sorry for that. The slides which I prepared about result interpretation seem somehow be not in the list for me. But please bear with me. I will find the presentation which I have prepared. No problem. Oh, thank you for your understanding. Thank you very much. There's a little bit, we have a little bit of problems with file sharing, and this is creating a little bit of trouble for me. So you can maybe stretch your legs and yeah, I think I have found it. So we have this just a short break. I will be, please bear with me, and I hope that I can show you what I actually prepared for today just in a moment. But you can write your questions in the chat. It's oh yeah. Please bear with me. It is loading up, but very slowly. Yes, you will receive the slides, no problem. Okay, I think I found the right presentation, which I actually wanted to show to you today. So I just need to scroll to oh, these technical problems are very disturbing, but bear with me. You got a little bit of break from my talking, and Now I am on track again. So we have been here with markers and their importance for analysis of your results. And now we are diving into this topic, which is of very big importance, that calculate an apparent molecular weight of target proteins are not always the same. You're probably aware of it, right? So, example, look, this is a protein of 751 amino acids, calculated molecular weight 83. But what you see on a gel, it's actually much smaller. Is it a correct protein I'm detecting? Actually, yes, because PSA-A, which is photosystem 1 protein, is very hydrophobic and it migrates faster. It will be apparent molecular weight will be lower. And there can be different factors which will influence protein mobility on SDS gel. So processing, of course, the protein can be shorter by 10, 20, 30 amino acids. Post-translational modifications, amino acid composition can change mobility by 10 kilodaltons. And also aggregation, adding of epitope tag, and also SDS page conditions. So please be aware of that and check it before you start doing your Western blots. And also when we have this ideal Western blot result, when we are purchasing an antibody from a company, there should be data showing that the antibody has been actually validated. And this is a perfect situation here. We have a band in a wild type, there is correct band is gone in a mutant, in a knockout mutant, and then we have an overexpressing mutant which is showing a band of a target protein. This ideal situation is, however, not always the case. And why sometimes an antibody detection of recombinant target protein is not a confirmation that the antibody is going to detect endogenous protein. If you see these data in product information sheet, please be aware that if it is a thick band coming from the recombinant protein, it does not mean that the antibody is going to detect endogenous protein with the same strength. In some cases, antibodies will detect only recombinant protein, but not endogenous one. And here it is a real example. So the endogenous protein band will be having much weaker intensity than the band from the tagged target protein. Please be very careful, and oftentimes it is thought that antibody validation on recombinant protein is automatically confirming that the antibody is going to detect also endogenous protein. This is not true. Antibody is not always recognizing target protein in overexpression mutant. Here is such a situation, and the band should be coming here, but not in this case. We have antibodies recognizing only target protein in a wild type. This is correct, but in the overexpressor, all this band pattern means that the antibody is not detecting overexpressed PIP2 protein. The band should appear first here at this molecular weight. Of course, addition, as you know from my webinar part 1, addition of urea can unfold the protein and maybe facilitate the detection. And also, sometimes we have situations when a band of target protein is not gone in the mutant. This is absolute nightmare. You see here the example of PIP7. We have been trying to develop an antibody to transcription factor 7. Unfortunately, we have this beautiful band. This is a result provided by another laboratory. And what to do in such case is what can be a reason for antibody recognizing protein of the similar molecular weight in a mutant. How is it even possible? There is a limitation of this method because what can happen is off-target binding. It can be incomplete mutant also, gene compensation. The list is long, presence of isoforms also. And the reason for the band being visualized also in a mutant is that, as you know, a band on a blot contains hundreds of proteins. And if another protein shares the epitope, the antibody will bind and there will be a band also in the mutant. What you can do is, in such case, to run adsorption protocol, which I have showed you on the previous slide. So if we don't have mutants or overexpressors, what can we do to be certain that the antibody is binding correctly? So we can have other approaches, like for example, fractionation. In this case, there was an antibody to an enzyme called PTOX, and this enzyme is localized in thylakoid membrane. So as you see here, the fractions were analyzed and the fraction of stroma was a negative control and the band was not visualized. Of course, there are certain situations where we cannot obtain viable mutants. And in such cases, we have to use other methods to confirm that we are detecting a correct band. Even if you are using commercial antibodies from established catalogs, it's good to include controls. Other methods which can be applied, it is also MS and immunoprecipitation and mass spec, and also sometimes we can use, if we have two different antibodies to the same target, we can see if the binding is at the same molecular weight. But not often we can have two antibodies to different parts of the same protein. And how to be sure that the antibody is binding correctly? Here is the checklist for you for the future. So you need to know if the molecular weight is correct, if the protocol is optimized, if controls are behaving as they should, and of course linear detection range, as I showed you. You need to choose correct chemiluminescent detection reagent. You can also use other approaches. And also it is very important to have, of course, reproducible results. So this is your checklist for the future. And if you have experienced the issues like poor results, wrong molecular weight of a target protein, lack of reproducibility, worse result with each blot, I have a solution for you, which I hope is going to be helpful. I would like to suggest that you're consistently reporting your Western blot experiments. This is a template which we are using when we are receiving results from laboratories which are testing Agrisera antibodies. So all these parameters are of crucial importance because, as you know, only a small change in a protocol can have a big impact. So you need to know exactly what has been done on each blot to be able to draw conclusions and improve your results in the future. So this is also the information which antibody providers should provide together with data for a given antibody which they load up to the website, include in product information sheet. And as you see here, we are covering in this template everything. Extraction buffer components, incubation time, blocker which is used, and it starts everything off with a load. Please do not load on volume. Remember it for the future that loading on volume can lead to results which will be difficult to interpret. And this slide I have already talked about, so I will skip it. And I have received this question: how to get an intense signal from Western blot using a weak antibody? You have been probably wondering about it yourself, right? So you need to ask yourself following questions. Did I use correct sample type? Instead of a whole leaf, maybe I should use a part of it, or maybe it should be another developmental stage of the leaf. is a protein of low abundance. Maybe the fractionation would be necessary to concentrate the target. Did I load enough protein per well? Was the transfer condition optimal? Were primary antibodies purified? Detection reagents, were they appropriate? All this is optimization. So I would like to encourage you to, instead of running blot after blot after blot, that you go back to the desk and use the template which I show you and analyze your results. This all starts with abundance of a protein which you're aiming to detect. This is absolutely crucial. So let me show you the example of optimization. Well, one could say, a lot of background bands, this antibody is not really that good, right? Look what happened. This is after optimization have been done. And look, what was the difference? The only difference in the protocol was amount of protein which was loaded, 20 microgram versus 2 microgram. I expect also that one could change incubation time to do it 1 hour room temperature instead of overnight to complete the experiment faster. So please remember that with these optimization steps, you can really improve your results. So record a correct band first, and then do not multiply conditions. If something goes wrong, analyze your conditions or contact me. And here I would like to present you with a protocol which we have worked out for antibodies which are just produced. We don't know if they are working well or not. And we are using, of course, correct samples, high protein load per well, and also we are using very concentrated primary antibody and incubated overnight. As you may guess, this may lead to increased background, but we will see with this protocol, with too much of everything, that a correct target protein band is going to be recorded. This is what we are after. The background, as I showed you, can be optimized later. And also, if I have a little bit of advice for experienced Western blot users, so look over your protocol. Do not use the same protocol again and again and again. And if you're still having problems and your blots look like that, don't be shy. Contact Agrisera. I will be happy to help you. And here we have common misconceptions about the antibodies. So I bought several antibodies from Agrisera, and therefore I think that they are going to work just fine in my setup. What do you think? Is it true or false? Think for yourself. The answer is coming. False. You cannot assume that it is every week I receive messages that this antibody worked with this sample, then the other antibody has to work too. No, this is false, because each protein antibody pair is unique. And the antibody works for species A, so for sure it's going to work for species B. True or false? False. You cannot assume it without sequence analysis. It's a lot of antibodies and time wasted because of this assumption. The same protocol can be applied for all antibodies. I think you answered directly false. This is not true. This antibody gives a good signal in Western blot, so therefore it should also work in other techniques. Is it true or false? False, because each technique requires specific validation. Another epitopes will be exposed in immunohistochemistry, and therefore you cannot assume that. You can read later about these common misconceptions, and also we have a technical blog where we are sharing the tips, and the posts to technical blog are included in our newsletters, and I warmly welcome you to sign up for the newsletter. I think you maybe already did signing for this workshop. And this is very important, how you're citing the antibodies. You probably have experienced it, that it is very common that the product number and supplier are not given in the material and method section. And this is actually due to lack of worldwide standard how to cite antibodies. And unfortunately, this is also contributing to the fact that more than a half of antibodies used in research cannot be properly identified. So to increase the reproducibility, please remember to also to cite the antibodies in that format. And of course, it would be nice to include, as you now have learned, it is good to know also what parameters have been used for the specific Western blot protocol. So this will help us to increase reproducibility, but also this would help you to save your precious time in the lab. And we are having some resources which I wanted to share with you. So educational posters you will find on our website. You can get hard copies or you can download PDFs. If you are looking for webinars and meetings to go to, then please visit this plant science event calendar. And also, if you are needing some pictures of model species and pathways, then you will find them for free on our website. So this is our gift for you. And also, if you want to, the last poster is covering very well this whole workshop, which I've presented to you. So if you would like to obtain it, you can do it on our website. You will find the link here in the presentation. Reminder about the 100 free antibodies. And now we are coming to an announcement of the winner of the quiz, which we had in the first workshop. This is Antea Mariani from University of Rome, and warm congratulations to you. And if you would like to also be a winner, there is a chance for you today. I will share the link in a chat in a moment, and please participate. You can do it until end of this week, and the winner will be announced next week. And I would like to thank you for your attention. And of course, I'm welcoming questions. So let's see what happened on the chat and which questions we have here. Oh, I see quite many. Oh, thank you very much. I really like that you are so active and you are providing me with so many questions. Let me just share the link to the quiz first, because I would like you to get the chance to win the antibody. And also that you will get the QR code is here, so you can just make it, use it from the screen, but I will also paste it in the chat. So let's dive into questions. What are your views on the performance advantages and limitations of solid phase dry protein transfer using the iBlot device? The efficiency of the transfer is going to depend upon biochemical properties of the protein, but also, of course, upon the equipment which is used. And high molecular weight proteins will be having better chance for transfer in wet type of transfer. But I wouldn't criticize one transfer over the other. And I would do like this, that if it is not successful, I would try, if dry transfer doesn't work and dry transfer requires more technical skills, then I would move towards the wet transfer and try it. I cannot criticize the specific devices, but I have seen situations when rapid transfers were working very well, but also in other situations they were not. So it is many parameters here, so I'm sorry that I cannot maybe give you a better guidance here. But I hope also that what I have presented will help you in the future to solve your Western blot puzzles. Second question, if a PVDF membrane has dried out, do you still recommend rewetting it with 20% methanol before proceeding? Also, if the membrane dries during imaging on the iBride and I need to rewet it to attempt detection again, what would be the best way to do it? Yes, you can use methanol, but also ethanol if you would like to use less toxic reagent. And we should actually keep the membrane from drying during imaging. So I would wash it. And during imaging, I wouldn't reactivate it again. I would just wash it, and as you saw on one of the slides, you can develop the reaction again. So that would be possible. And now we're coming to the second question, or if you have any questions which are left, for me, you can also mail them. I can also answer them later. Let me read for you the next question. The quiz QR code I have just pasted. You will get recording and the slides. I don't know which link is not working. Is it the link on our website, on the form, which is not working? Please specify it. Another question. I also have another question about Ponceau staining. Some protocols use Ponceau prepared in acetic acid, while others use TCA. Do you have recommendation on which is preferable, or situations when one is better than the other? Well, I would stick to the protocol which is suggested by provider of the Ponceau. This is what I like to do. I always ask suppliers of reagents for help and for providing me with suggestions about the protocol. So this is what I would do. You can also use Kumasi for reversible stain, and I have covered it in a part 1 of this webinar. So even with a technical difficulty, I managed to be on time. And please tell me which link you referred to, which was not working, to the recording and slides. I guess you referred to part 1, because after this workshop today, you will receive a message and with a recording and PDF of the presentation. I encourage you to participate in the quiz, and the winner will be announced next week. Link shown in the workshop, okay? Okay, let me share the recording link with you. So here you will find the recording of the former workshop, and also until end of the week there will be recording of this workshop, and you will be notified by mail. So thank you very much for your time today. I see one more question. Thank you for this precious webinar. You are most welcome. What's the best way to store developed membranes? I usually do it in plastic sheets and in the dark, but I notice that over time the signal fades out. I'm usually developing with alkaline peroxidase. Well, unfortunately, it is good to record after development directly, and unfortunately, if you develop with alkaline peroxidase, the signal will fade with time. And I think it does not matter if you store it in darkness, or we are storing the blots in the lab, not in the darkness, and the signal is still fading. So document by making pictures. It will be better. So if you have any more questions, please send a mail. And I don't want to hold you up more. And I thank you for your time today. And it was a pleasure to share this knowledge with you. And I wish you good luck with your blots. And please do not hesitate to write to me. I'm always eager to help and learn also from your mistakes. So thanks a lot and good luck.